Thank you, Helena. I'm really indebted to you and to many people at the ADRC and to the Batey Foundation, Batey family for their support. It's so good to see everybody, and I think we are so motivated by the interactions with our patients and our research participants. So bear with me a little bit so I can find the presentation. Okay. All right, so you know, a lot of this research would not have been possible without the support of many people at USC. Helena has been instrumental in my own career development at USC. Lon Schneider and others have been outstanding mentors. So I have to say that this research is all homegrown. It's built on collaborations between different schools and departments, and I am honored to be part of this division. So as Helena mentioned, I'm interested in apolipoproteins, and ApoE4 is really the elephant in the room when it comes to Alzheimer's disease. In at least the white population, around 20 to 25% of people carry one copy of E4, about 3 to 5%, 2 copies. And that's quite an enormous allele frequency, speaking to the importance of this genetic variant in our ancestry. Now, our ancestors perhaps did not live to become 80, 90 or above, and there is a price to aging. Those who carry one copy of the gene have a 3 to 4-fold increased risk of dementia, Alzheimer's. And those who have 2 copies, if they live to become 80, and some of them do, they will have a 50% chance of having Alzheimer's disease, dementia. So ApoE4 is a really big issue when it comes to Alzheimer's. We still don't understand the main mechanisms of how ApoE4 leads to disease. We don't have any solid recommendations on what diet, lifestyle factors. We don't have any drugs that can target ApoE4. And that's exactly the focus of my research. I'm just gonna, because I only have 10 minutes, I'm just gonna give you, the spiel of what we have been doing over the past decade. So we know that ApoE4, by virtue of being a lipoprotein, affect brain lipids. We have shown that ApoE4 carriers before the onset of dementia have lower omega-3 brain delivery. But does that mean that omega-3 supplementation works? We don't know, and I'll share a few slides at the end toward that end. We know that ApoE4 carriers with dementia have an increased measures of brain inflammation. Can we inhibit that? And will that translate into clinical benefit? And finally, how can we assess ApoE function in the brain? And we have developed an instrumentation to be able to measure ApoE HDL with the support of the Batey family. So let me just give you a little bit of background why omega-3s. The most important omega-3s include EPA and DHA. They're largely consumed from fish, and they make a big bulk of the lipids found in the gray matter of the brain. They have anti-inflammatory properties, and there's so many epidemiological studies that suggest those who consume more omega-3s have less incidence or less risk of developing Alzheimer's dementia. However, this is the conundrum. If you look at clinical trials that tried omega-3 anywhere from 6 months to up to 2 or 3 years, they found that DHA supplementation has largely been ineffective in patients with dementia. To make things even more complex, even those with small trials that looked at AD prevention, the findings were inconsistent. So what does that mean? Is it really just a biomarker? Does it have any role in the disease process? So we got interested in this question around 2013, 2014. So one of the key studies that really stuck my attention was a paper by Joe Quinn, published in JAMA in 2010, showing that 2 grams of DHA for 18 months had no effect on cognition in patients with mild AD. This is very clear in those who are ApoE4 positive. If you look at those cohorts, you'd see the scores did not change at all in E4 positive. And the higher the score, the more mistakes somebody is making on this test, the ADRC. However, the E4 negatives showed maybe a sign of improvement, but the point from this paper is that ApoE4 AD does not respond whatsoever to 2 grams of DHA over a period of 16 months or 18 months. So why is that? Why are ApoE4 carriers resistant to omega-3 supplementations? These are ApoE4 carriers with AD. So I am fortunate to be surrounded by outstanding faculty and colleagues at USC. And Stan Louis' lab at USC has quite an interest in inflammation in the setting of many diseases, including eye diseases, liver diseases, and cancers. So I went to Stan approximately two to 3 years ago, and I said, hey, look, we're interested in why ApoE4 carriers are not responding to omega-3 supplements. I know you have an interest in inflammation. Can you test these brains? And we got these brains from the Rush ADRC. Rush has one of the largest databases of brain banks in the world, and they are well phenotyped and broken down by ApoE genotype. Long story short, what we find here is that ApoE4 carriers with dementia differ from ApoE4 carriers without dementia. At death, those who died with dementia have sustained chronic inflammation as measured by all these lipid mediators. There's more inflammatory lipids and there's more anti-inflammatory lipids. The brain is stuck in this conundrum of trying to resolve inflammation, but the inflammation is out there. When we looked at the pathways that can cause this inflammation, we ended up with an enzyme known as calcium-dependent phospholipase A2. That's an enzyme that's activated in membranes and leads to digestion of lipids. So it can possibly explain why we see so much atrophy in areas of Alzheimer's disease brains. The atrophy that Ionis has showed us in a little bit, and some of the pathology of amyloid that Dr. Binkowski showed us. So over a course of 3 to 4 years, Xiaowei Wang, a very talented postdoc in my lab, looked at careful models from ApoE4 cells, ApoE4 mouse models to human brains. And this is a long study, but I'm just showing the conclusion, and identified that CPLA2 activation is underlying the digestion of membranes and inflammation that you see it in the ApoE4 model. We were very excited, and we asked, is that potentially a drug target? So to be able to move into a drug, it's a very challenging pathway. It's an expensive pipeline that requires all kinds of expertise. With the help of the Mesh Academy and many people within USC, I was introduced to Seva Katridge, who is a computer scientist at Michelson's at the main campus. Seva is a very talented computer scientist who can actually plug in a structure into a huge computer and identify in silico or hypothetically all small molecules that can, in theory, inhibit this actual protein. So we gave him the structure of CPLA2. We asked Seva, can you identify theoretically small molecules that can actually block the function of this protein? And Seva said, let's give it a shot. I'm excited. Alzheimer's doesn't have any medications. Why not? After 3 to 6 months, Anastasia, which is a leading postdoc in Seva's lab, identified 127 compounds who seemed to be safe, who seemed to cross the blood-brain barrier, and were able, at least in theory, to dock into the sites of this protein to inhibit it. Marlon Duro from my lab tested now all these 127 compounds, identified 7 of which indeed in the test tube inhibit CPLA2. Then Xiaowei Wang did these studies, and I know they may look complicated, but long story short, 3 of these compounds inhibit the inflammatory mediators that are driven by CPLA2. For example, prostaglandin E2 is a measure of inflammation, and you could see, for example, 50054, one of these compounds, successfully blocked PGE2. COX2 expression is, by the way, inhibited by NSAIDs like Aleve that you can buy over the counter. And as we expected, 50054 successfully lowered COX2 expression. These are downstream of CPLA2 activation. Once CPLA2 is activated, you've got all these enzymes which are pro-inflammatory activated as a result. So when we block it upstream, we block the downstream cascade. So the next step for us is, can we actually take these small molecules and make them into a drug? And that is not an easy test at all. I mean, we have been for the past 3 months trying to solubilize them. They're extremely hydrophobic or fat soluble, meaning that they can possibly cross the blood-brain barrier effectively, but they're very hard to make into a pill. So I reached out to Stan and I told him, hey, what does it take to make these drugs? And Stan looked and said, this is not easy. That's a huge project. Are you sure you want to go into this? And I said, let's do it. So with the help of the Batey, Gail and Ben Batey and Mike Vranos, we were able to get enough support to get these important experiments on the way. And I'm really, honored and grateful to both Mike and Gail for their support to help us develop a drug here at USC. So then the other question, because I started with omega-3s, is there any role for omega-3 supplementation in ApoE4 carriers before the onset of dementia? And let me walk you through some of the major epidemiological studies. So greater consumption of seafood from the KAID study from Finland, long-term use of omega-3 supplements in a cohort known as ADNI, the Alzheimer's Disease Neuroimaging Initiative, and higher levels of omega-3 in blood from the Framingham offspring cohort, all showed that ApoE4 carriers were eating more seafood, having long-term supplements, or have higher levels of omega-3 in their blood, has better cognition. Now that could be a biomarker. That could be something else that explains why they have better cognition. To answer this question, you really have to put it in the context of a randomized clinical trial. So first, I want to acknowledge Lon Schneider, who has been a fantastic mentor and a friend, and helped me design PREVENT-E4. PREVENT-E4 is a randomized clinical trial trying to understand whether 2 grams of DHA, the omega-3 supplement, versus placebo can have an impact in those at risk of getting dementia because of ApoE4. So the study started in around 2017. We were supposed to finish recruitment in 2020. COVID happened, delayed us by 1.5 years, but we finally did it. I mean, we really celebrated, went out as a team and celebrated on the day that we finished randomization. So we screened over 2,000 individuals all across LA and sent them test tubes to be able to collect saliva. The test tubes were being sent back to the lab. We lost a lot of test tubes because of COVID. Some of the samples were, thrown at the entrances because all the doors were locked for 1.5 years. We had to apologize from all of you and research participants to do some of these testing again. But finally, on May 1st, 2022, we reached that magic number of 364 and closed recruitment. The study has 2 more years approximately and will conclude, and we'll find out whether high doses of DHA in ApoE4 carriers without dementia can actually make an impact on blood DHA levels, brain DHA levels, MRI changes, or cognition. So the last part, and this is the last few minutes, is what does ApoE do? I mean, do we understand how ApoE functions? And ApoE, by definition, is an apolipoprotein, meaning that in the body, it will bind lipids like cholesterol and form a particle that we call HDL. Now, despite all progress in ApoE research, ApoE HDL in the brain has not been measured, and partly because it's not very abundant. It's actually about 1,000-fold less abundant than blood. And the way to measure ApoE in the brain is to obtain cerebrospinal fluid. And that's why we ask a lot of our research participants to be patient with us and to allow us get CSF during our studies. It's a very important fluid that helps us understand what's happening in the brain. So again and again and again, I'm really grateful to the Batey Foundation that supported us capturing a very fancy instrument that is extremely sensitive and can measure HDL particles. They raised a challenge. Ms. Batey asked us to match it, and the department came through, matched 50% of the cost, and we were able to purchase this instrument. This instrument works by putting an electric shock into the particle. The particle will be surrounded by a water coat, and then it will float. When it floats, The analyzer can measure its size and concentration. And we know exactly how HDL looks like based on the configuration of these particles. So again, I cannot thank all of you for participating in ADRC studies. These are 200 individuals. You could be one of those people that provided the CSF, where we actually measured these HDL particles using ion mobility. And the mean age here was 76. And you could see the cohort between males and females, Latinos and non-Latinos, and with and without cognitive impairment. So what did we find? In a nutshell, this was published in Alzheimer and Dementia 6 months ago. And what we found is those people who have more small HDL particles in CSF as a measure of small HDL in the brain did better on memory, global cognition, and measures of attention and executive function. Why is that important? It's important because now we have a target. Can we develop a drug that can increase small HDL in the brain? HDL is a functional part of ApoE. These are largely ApoE HDLs. And then this will lead the way for us to develop new generation of drug targets. So let me conclude here that in patients with ApoE4 and dementia, we have identified CPLA2 with the help of Mike Vranos and the Batey family. We are developing CPLA2 inhibitors. In patients without dementia, we just finished PREVENT-E4, a randomized clinical trial to understand whether high doses of DHA can help the brain in those without dementia. Using the lipid analyzer, we're now developing new biomarkers that can guide the future treatments that are aimed to improve the HDLs in the brain. And finally, I want to thank you all for participating in our studies. Without your generous support, we could not be doing this. I am grateful to Helena, to Lon, to Vranos, to the Batey Foundation, to NIA, to the Alzheimer Association and Alzheimer's Drug Discovery Foundation for all the support that they have provided. And, you know, this is fun. I love what I'm doing. I think it's quite exciting. It takes really a village of people, a large group of scientists to be able to do the research we're doing. We cannot do it without you. And I hope that one day we would come and say, you know, we did something, our drug reached the clinic, or our intervention is making an impact. At the end of the day, we want to help you, and you are helping us at the same time.
Thank you, Dr. Chu. Thank you for a very nice introduction. Hello and good morning to everyone. It really is an honor to be here today. As Dr. Chu said, my name is Maria Aranda, and I'm the lead for the Outreach Recruitment and Engagement Core. And this is the unit of the center that deals with providing information and education, mainly to the local communities. I'd like to give thanks also to Gail Batey and family for supporting this Appreciation Day event. You've all had front row seats to this premier event, and you've heard about the exciting and groundbreaking studies that are being conducted by the esteemed scientists at the USC Alzheimer's Disease Research Center. And I'd like to thank our speakers today for excellent presentations. I have a few take-home messages today. First of all, as has been said, but I also want to echo this, we are eternally grateful for you. None of this exciting work would be possible if it weren't for you, our study participants, and also study partners. You've given yourself unselfishly to participate in the studies highlighted today, but many other studies as well. Our work is paving the way for a more accurate diagnosis, understanding how the disease process works, what are the mechanisms of action that should be targeted in the future for better treatments, a better care, and hopefully a cure. So again, we thank you immensely and hope you continue with us. As said by one of our participants earlier today, each of you is giving a piece of a piece of a piece of data. And I couldn't have said that any better. Second, when you think about brain health and memory research, think about the USC Alzheimer's Disease Research Center. This center really is the hub of scientific discoveries, as you've already seen. And this is the case for generations to come. And lastly, you are our center's ambassadors. So what does that mean? We ask you to promote, to encourage, to cajole your friends, your family members, your neighbors, co-workers, congregants from your house of worship to engage with us and to join one or more studies that we sponsor. We know that you have many studies to consider, but you do have information at your fingertips today. You have in your packet of information a list of studies that are currently active or pending in our center. We ask that you review those. If you're interested in being a partner and volunteering for any one of those studies, just let us know, provide us your contact information. In closing, you do also have an event evaluation form in your materials. We want to make sure that we hear from you about your impression of today's webinar. Also, maybe ideas of future webinar topics. And again, if you're interested in participating or want to send somebody to us, feel free to add the contact information on the last page of that evaluation form. With that, I thank you, ambassadors.